prazer estar aqui e eu gostaria muito de dar essa aula em português, mas é impossível com o meu vocabulário de menino de 10 anos. Então eu vou ter que falar em inglês. Realmente vai ser impossível em português. Então, what I want to do this afternoon is I'm going to tell you first a little bit about how I think about problems in immunology. And then I'm going to tell you about, summarize our work on HIV and this problem of the HIV vaccine and why it's so difficult. And then I'm going to tell you just a little bit about something that I'm excited about that we've just done, which involves a collaboration with Brazil, actually, on the problem of Zika. Okay, so I'm going to ask the man in the booth to change the slides when the time comes. This diagram tells you three of the key features of the immune system that make it different from the brain, that make it different from kidneys and other organs. And it provides a way, it's important because it provides a way of thinking about what is unique about the immune system. And there are three features that are special. One, it's very diverse. And the second is that it has a great deal of specificity. And the third one is that it has memory. And all these three things can be accounted for in this simple diagram, which is a theory that was put forward by Burnett many, many years ago, before any of the details were known. So what the Burnett idea says is that the immune system is composed of cells, that each cell is a clone that has an individual receptor. So that's shown here in different colors. And the specificity of that clone of cells comes from a receptor on the surface of the cell. Okay? And then memory in this very simple model comes from the fact that when a pathogen, a virus or something else comes in and comes into the system, it selects for a lymphocyte, a specific lymphocyte, which is then expanded clonally. And then when the pathogen comes back, well, there's a expanded clone, so it reacts faster and better. And that's the basis, in this idea, that's the basis for vaccination. Now, if you could show the next slide. There's, próxima, forward. There's something that Burnett didn't know about when he put forward his thesis. And that's this phenomenon that was discovered a few years after Burnett. And that is that when we are immunized, or any animal is immunized, there is a rapid increase in the affinity of the antibodies in our serum in a matter of 2 or 3 weeks. And that cannot be accounted for by the Burnett idea. And it turns out that what we've learned over the last decade, or last 20 years really, if you show the next slide, is that the lymphocytes, the B lymphocytes in particular, the ones that make antibodies, do something very unusual when they go this clonal expansion. They do something completely strange, unexpected, and it's something that explains why, in fact, adults have many B lymphoid cancers. And that is that instead of a monoclonal expansion of a monomorphic group of cells, the immune system turns on an enzyme that mutates the antibodies, that mutates the receptors, and generates a whole group of cells that are in fact very closely related to each other, but differ by small mutations. And then it selects, the immune system selects for the best clone. Now on the next slide, you can see the basis of this reaction, what we've learned in the last 10 or 15 years actually, is that what happens is that there's an enzyme called activation-induced cytidine deaminase, and what it does is it deaminates cytosine, creates uracil, and that's a mismatch. Okay, so then that's a mismatch in DNA. It's recognized by the normal cellular enzymes that recognize damage, and this damage is then converted to somatic mutations, gene conversions, class switching, which is important for antibodies, and can result in chromosome translocations, which is really what we've studied, which lead to B-cell cancers. So mistakes by this system lead to most B-cell cancers in adults. Okay, next slide. Now, the enzyme that does this, it's not like a restriction enzyme. It's not site-specific. It can't be, because if it were, it would always make the same mutation. And what you want is diversification, not the same mutation. So it's not really sequence-specific, but it has some preferences, and it prefers these areas. These are the parts of the antibody molecule that recognizes the pathogen. That's where you want the diversity to be focused, and that's where the enzyme focuses. And if you just show the next slide, this happens in a microanatomically defined compartment in the immune system that we've been studying called the Germinal Center. It's something that's been known by pathologists for many, many years, 100 years, and it exists in lymphoid organs. And it has two zones, a light zone shown here, pink because it contains the antigen, and a dark zone where cells proliferate. And the next slide just shows a model. I'm just going to summarize a lot of work over the last 10 or 15 years again, where we've learned something about how affinity selection occurs in these germinal centers. So the germinal center is separated in two zones, a light zone and a dark zone. In the dark zone, next slide, the B cells divide. They divide and they mutate in this compartment. And then this is very dynamic. It's not a static organ, but it's in fact an organ where the lymphocytes are moving around. And after they finish dividing and mutating, they massively migrate to this other light zone. And the light zone is where they meet the antigen. So they have a new receptor that they've mutated, and they're going to test it in the light zone. They test it by interacting with the antigen. The next slide. And there, they also receive communication from T lymphocytes. And the T lymphocytes are, in fact, responsible for detecting the antigen that the B cells have taken up. And next slide. They then, seeing the B lymphocyte that has picked up the most antigen, will drive them back down into the dark zone, where they repeat this cycle. Next slide. So then they divide again. So the cell that has divided, mutated here, moves here, picks up the antigen, meets the T cell tells it, go back if you're a good one, and repeat this cycle. If you are not selected, you die. And you do this many times. And because you do this many times, you can select for the cell with the highest affinity. So that's how that whole process happens. Next slide. Okay, so I want to turn now to HIV, and this will be relevant. What I told you will be relevant as we go along. So this disease is a disease that, although it's preventable, we all know that it's preventable, it affects many, many people. And the incidence rates have really not changed over the last 5 years. In fact, they've increased in some parts of the world. So although it's preventable, and although we have really good drugs, incidence hasn't changed. People keep getting infected. Next slide. And the problem here, a big problem, is that many people who are infected are not on drugs, and so they continue to transmit the disease. So here, the number of infected people, here are the people that are diagnosed, so about half of them are diagnosed. And then this is the number that get on to medication, and then this is the number out of the total that are in fact suppressed, that won't transmit the disease. So big problem that people don't take their medications or they don't get diagnosed, and so this is a continuing issue. Next slide. Okay, so we know this is a big global public health problem, and we know that 30 years later there's no vaccine and there's no cure. So the question is why? Why has this been such a difficult problem? Just say a little bit about that. Next slide. Okay, so the first problem, the first really difficult problem is this, is illustrated in this slide. So here you have the neutralizing breath of an antibody, of all antibodies actually, for all viruses. This is 100% of all viruses, and this is the neutralizing potency. And up until 2009, which is not a very long time ago, this was the best you could do with antibodies. These were the 4 best antibodies that were cloned in all laboratories all over the world against HIV in terms of their potency and breadth. And the problem is that it takes a lot of antibody, 10 micrograms per mil here, to reach any kind of reasonable level of coverage. That level of antibody is not possible to make by vaccination. So if that's not possible, and most vaccines are made by antibodies, then you cannot have a vaccine. So people were very pessimistic about vaccines because of this. The next slide shows you an additional problem. This additional problem is illustrated in this structure from the laboratory of my collaborator. It's a collaboration, in fact, of the HIV envelope spike. This is the only protein on the surface of the virus. And what is difficult about it is the blue stuff. And the blue stuff is glycan, it's sugars, and they are self-sugars. And the immune system is trained not to see self, because of course if it saw self, you'd be autoimmune. So it has to avoid self, and it has to avoid all this blue stuff. So there's very little that the virus is not covering up and making believe, in fact, that is self, that the immune system can see. There are a few spots. One of them is illustrated here. That's the Cd4 binding site. Cd4 is the receptor for this virus on the human T cell. And so this has to stay open so that Cd4 can get inside. Next slide. So in addition to having some targets, some potential targets like the Cd4 binding site, there are some good bits, some good things to think about in terms of optimism for a vaccine. And one of them is that it's been known for a very long time that a few people, just a few people that get infected actually make antibodies that are broad and potent neutralizers. So although antibodies to HIV had not been discovered to be broad and potent, it was known that people could do it, that they had it in their serum. And it was also known that this was a strange immune response, because unlike the slide that I showed you right in the beginning where the affinity goes up very quickly, these people took 2 or 3 years to develop this kind of immunity. So this was an unusual kind of immunity happening in a few individuals. And what we wanted to do in the lab, next slide, was to find out what these people had. So we had to develop a technique in order to clone the B cells from these people, the antibody-producing cells. And this was the work of a graduate student, Johannes Scheid, and a postdoctoral fellow, Hugo Mouquet, who is now a faculty member at Pasteur. And what they did is they said, okay, look, we know what the protein is that has to do this. We know the cells, they're the B cells, so we can label this protein and use the labeled protein to find the few lymphocytes that are circulating that actually recognize the protein, and then clone the antibodies and reproduce them by molecular biology. Okay, so next slide. So that actually worked, and it worked very well, in fact. And this kind of method has been adopted by many laboratories and been used to discover antibodies. And what happened was that we now have antibodies that are far, far more potent than what we had before, 3 orders of magnitude, and they are also very broad. That is, they see many, many strains, most strains of HIV. The other thing that happened as a result of this work, next slide, is that we discovered that, in fact, in addition to the Cd4 binding site, there are many other targets on that viral spike. Although it looks like an antibody can't get past all those glycans, in fact, they can. And they do it in a rather special way, which involves the Germinal Center, what we talked about earlier. Next slide. So one of the major surprises, actually, when we did this work, when we initially did this work, was the finding that the antibodies were very heavily somatically mutated. So here are a list of these broadly neutralizing antibodies that were discovered. These are the framework regions of the antibodies, not the loops where I said that AID should concentrate, but in fact the structural bits that should not be mutated. And you can see that these structural bits are peppered with mutations. In fact, some of these antibodies have 100 mutations in a 300 nucleotide V gene. That means that they can change every single amino acid in the variable gene. Very, very unusual. Next slide. So the mutations are essential. So if you here are binding SPR traces for several antibodies in their mutated state and in their unmutated state. If you take away the mutations, they don't bind. And more importantly, these are viruses that are neutralized by the antibodies. You take away the mutations, they don't neutralize either. So the mutations are there, very unusual and essential. Next slide. So why do you need so many mutations? These loops here are really not a big part of the structure, but you have all these mutations. Well, the reason is, well, we've done a lot of structural work, primarily with Pamela Bjorkman at Caltech. Next slide. And what we and others who've done similar work have found is that these antibodies are not binding in the traditional way. They are using different bits and pieces in order to get past all those glycans, including here one of the structural bits and not the loops. Next slide. So when we saw this, we said, okay, well, look, this cannot be the product of a normal germinal center reaction where you get a couple of rounds of this, you get affinity maturation and selection. In fact, what must be happening is something unusual. Either cells are going out and then coming back into germinal centers, or there is a persistent, prolonged germinal center reaction. How would this happen? Well, it turns out that others have in fact discovered it by doing some incredible experiments in people, prospective studies, where they've taken an individual who's infected with the virus and then followed for 2 or 3 years and cloned the virus and the antibody at all the different stages. Next slide. So this is an animation slide to illustrate this. The virus protein, the cell surface protein, is shown here. That's the spike. Virus comes into an individual, and the individual makes an antibody. Next slide. Now, that antibody will select against that virus. It's specific for that virus. It's not broadly neutralizing, but it's specific for that virus. And because HIV is mutating, that antibody selects for a variant. Next slide. Now, that variant is resistant to the antibody, but the antibody chases. Next slide. And then selects against this one. Next slide. And then another one escapes. And the antibody chases again. And you can keep on going. Another one, and another one, and another one, and another one. And over 2 or 3 years, next slide, you get broad neutralization. You get an antibody to evolve through mutation and selection against what turns out to be many different viruses, something like this. Okay, so next slide. So if that's true, how do you copy it? How do you make a vaccine? Well, you cannot possibly expect that the immune system will do this with a single antigen, because if you put in a single antigen, you're going to get selection, you're going to get affinity maturation, and it's going to be done, and you're not going to go all the way to this. So you have to imagine that you would have to produce something akin to what happens in the infection. And you might be able to do that with a variety of different antigens, a sequential, a new kind of immunization. Next slide. So how do you test that? Well, you would have a great deal of difficulty testing that in a monkey or a person or in a mouse, simply because of the number of variants and the number of antibodies involved. And so what we've done, together with protein engineers at Scripps, in our lab, is to make mice that carry the precursor of the antibody we want to get. So we know what we're starting with, and as somebody told me, you can't end unless you start. We know what we're starting with, and we know where we want to get in this defined, simplified system. Next slide. And the antibodies we did this for are two. This one, 1074, and this one, 3BNC117. This one sees the Cd4 binding site, and this one sees the top of the molecule. These two antibodies are also the same antibodies that we currently are testing in the clinic. Next slide. So the protein engineers provided us with a series of antigens, some of which were very open structures. The reason we wanted to do that is that you can imagine that getting in past all of those glycans would not be a trivial thing, but maybe we can train the antibody, in fact, through sequential immunization to go from an open structure to a closed structure. Next slide. And that's exactly what Emilia Escalano did in a series of experiments, starting with very open structures and then moving into more closed structures. And this was guided, this kind of immunization was guided by simple ELISAs. Next slide. What happened at the end is that we got broadly neutralizing antibodies, and that was the first time that anybody produced broadly neutralizing antibodies to HIV in any system, actually. And those antibodies were highly somatically mutated, so they were exactly as we predicted, that they would be highly mutated and they would only occur, in fact, with a sequence of immunogens. Next slide. So this is where we got to with this mouse experiment. We ended up using 9 different immunogens in order to achieve this. And we did it in an engineered, simplified system. But it was a demonstration that, in fact, you can do this, because before this, it wasn't clear that you could in any system. Next slide. But there are lots of caveats with this experiment. It's a highly simplified model. Mice are not humans. I don't really believe that, just so that you know. The knock-in, it's really biasing the system in a very strong way. Only the precursor frequency in particular is highly abnormal, and only a fraction of the cells are responding. And for the 2 different kinds that we made, one of them we were able to get most of the way there, but not completely. And the other one, we did get all the way to broadly neutralizing antibodies, but they are still not as good as the ones that we got in the humans. All right, so next slide. What do you do then? We have these antibodies. We're working towards trying to develop ways of making this vaccine by the sequential immunization, but clearly it's not a trivial issue. It's something that's going to require quite a bit of additional work. And so we've begun to explore what we can do with the antibodies clinically. Next slide. And there are really 4 things that you can imagine that you would do with the antibodies in the clinic. And they are passive protection, that is, transfer the antibodies and see if they will protect people from acquisition. Post-exposure prophylaxis, chronic therapy, and eradication. Okay, so next slide. Why would you do this? Antibodies are expensive. They're more difficult to produce than small molecule drugs. And maybe they're inconvenient because they have to be injected. But antibodies do things that drugs don't do, at least so far. And one of the important things that antibodies do that drugs don't do is that they connect the pathogen with the host immune system. And they do it through their Fc domain. So they can clear the virus from circulation, they can kill infected cells, and they can create immune complexes that activate Ralph's dendritic cells for antigen presentation. So they have a series of functions that drugs don't have, and that is why they are also useful, particularly useful in treating inflammatory diseases and in treating cancers. Next slide. So we've done experiments in humans, and I'm going to show you first just experiments in macaques, just 2 recent experiments that we've done, and summarize the human clinical work. Okay, so for these 2 antibodies. So next slide. So the first thing is passive protection, and this is the thing that's furthest along and getting very close to large efficacy trials. Next slide. So the model for this, next slide, is macaques. And it's macaques and a virus called a SHIV, which is an adapted HIV. And what this experiment does is to inject the animals with antibodies and then challenge them every week with a low dose of HIV. Just like people. People take a long time to get infected, actually. Most people don't know this, but it's really hard to get infected with this virus. You have to try hard, and in fact, people do. And so what we do in the monkeys is give a low dose many, many times. All right, but we pre-treat them with antibodies. Next slide. And if you don't do anything, what happens is they become infected. It takes them some weeks. This is weeks here. It takes them some weeks to get infected. Next slide. If you inject the antibody once, depending on the antibody and the form of the antibody, so it can be a natural antibody, that's this dotted line, or it can be a modified version that is long-lasting, a particular mutation that makes it long-lasting. You can see that a single injection can prolong acquisition time very significantly. And for this one here, up to 36 weeks. So one injection takes 36 weeks to get infected for these macaques. Next slide. So on the basis of this, human trials have been started, not by us so far, but by the NIH, where they're going into Africa and also in the United States and giving people injections of antibodies periodically to see if this will prevent infection. Next slide. All right, but that doesn't do something to the host. It's simply prevention. Drugs can do that as well. Next slide. So what about the host immune system, and do the antibodies do something to the host? This paper was just published, where we treat infected macaques with the antibodies. And so, next slide. The model here is a SHIV infection again. What happens when you infect monkeys is that they get viremic, they get a lot of viremia, and then they stay viremic for many weeks here. At that time, at the same time, they are dropping their Cd4 T cell counts, which is what happens in AIDS, and then out here, after many, many weeks, they in fact die from AIDS, an AIDS-like disease. Next slide. So what we did was to infect the monkeys and then treat them with antibodies in this gray bar. So the antibodies are there for this period of time, and they're coming out of the system. Monkeys get their viremia suppressed. So we don't see viremia because the antibodies are capable of suppressing the viremia while they're there. But of course they have a certain half-life and they're coming out. You can see, like, for example, in this monkey here, the antibody goes away and the monkey becomes viremic. But then something strange happens, which is that they drop their viremia. Viremia goes away. Okay, so that doesn't happen in a normal circumstance. Something happened there. And it happened not just in this monkey, but it happened in these other monkeys. Next slide. And it happened in about half of the monkeys that they were completely suppressed in terms of viremia. And this is weeks after, and you can see this is 50 weeks, that sort of thing. So for a year, no viremia. The other half could be broken down into ones that developed AIDS, like this one who dropped the CD4 T cells, the red line, and these two, which have very low levels of viremia. They're controlling their viremia and maintaining their CD4 T cells. So those are also so-called elite controllers. Next slide. That doesn't happen with drugs. So if you do this in people or you do this in monkeys, treat them with ART, this does not happen. So this is something that happens with antibodies but not drugs. Next slide. And exactly what happened. So, the truth is we couldn't figure out what happened for a long time. We thought that this would probably be due to immunity, to T cells, in fact, that were controlling the virus. But we could not find them. And so we did the following experiment. We waited here, you see, a whole year. And the viremia, so it goes up, and then it goes down and stays suppressed. And then we injected antibodies to CD8 T cells that depleted the CD8 T cells. And when we did that, what happened was they became viremic. And then they got their CD8 T cells back, and they controlled viremia for another whole year. And we repeated the experiment. And they again became viremic. So these monkeys are controlling their infection for a couple of years, and they're doing it with CD8 T cells. So what happened? And what happened in the monkeys? Next slide. Well, we don't really know exactly what happened, but we have an idea, and it's sort of illustrated here. When animals are not treated, the infection essentially destroys their immune system, so they're unable to develop this kind of immunity. If you give drug therapy, what happens is the viral cycle is interrupted, and there is no antigen. There essentially is nothing for the immune system to respond to. With the antibody therapy, what happens is the cells are there, they're producing virus, and the antibody is capturing the virus, making immune complexes, and then dendritic cells and other cells in the immune system can react to the antigen. So we believe that that's how that CD8 T cell response is created, and that's not something that you can do with drugs. Next slide. Okay, so in fact, we've done experiments in humans. Those are macaque experiments. We've published the human experiments. And in humans as well, we believe that there are immunologic effects of the therapy. Next slide. Okay, so these are, I've shown you preclinical studies. I want to tell you a little bit about where we are with the clinical studies as well. I'm just going to summarize it in one slide. Next slide. Okay, so we started this cloning effort in 2009. We cloned the first antibodies in 2011 and 2012. The Gates Foundation helped us to produce the antibodies for clinical studies, for experimental medicine studies in people, small studies, which we began in 2014 and in 2015. We've done these with individual antibodies. We know that they're effective, we know they're safe, and we know that the virus escapes from the single antibodies just like it escapes from single drugs. And we've just started combination studies, putting two antibodies together to different targets that are non-overlapping, just like drugs. And the results look really, actually terrific for this combination. Now, what our plan is to use the antibodies for prophylaxis, really. And we're going to do that in Africa. We have formulated the antibodies so that they are injectable subcutaneously. And the target population is young women. So in Africa, taking pills is somehow means you're sick. And so young women, who are the people who most need this kind of protection, don't take pills. They will not take ART for prevention. But they do like birth control, and they come to clinics every 3 months for subcutaneous injections of birth control. And that empowers them because they don't get pregnant all the time. And so our plan is to formulate the antibodies so that we can inject them every 3 months. When they come in for birth control, we're going to give them the antibodies. And that'll be the initial clinical study. It'll involve a couple of thousand people, and we are in the midst of planning to execute that study. Next slide. All right, so what we did was we started with people that had high levels of antibodies, and we discovered something about those antibodies, and we discovered something about vaccines and why it's so difficult and so on. We wanted to know whether that paradigm was something that is translatable to other diseases as well. And so we collaborated with investigators here in Brazil and in Mexico to try to do this for Zika. Okay, so next slide. What we did was go to this Pau da Lima, in Salvador, with the help of Albert Koh, and we screened 300 individuals for reactivity to Zika virus. This is a place that had an outbreak. We also did this, next slide, in Mexico, in this place, Santa Maria Mexitalia, with collaborators in Mexico, and screened an additional 100 people. Next slide. We looked for neutralization in addition to binding, and we got a bunch of people that were high neutralizers. Next slide. And then we actually collected their blood. we collected their blood. We came here, actually, collected their blood from these few people, and then did the same experiment that we did for HIV. That is, we used the Zika protein in order to sort single lymphocytes and then clone the antibodies. Next slide. And we got a surprise, something that I was totally not expecting, which is here are the number of antibodies that we cloned from each individual. The slices are clones. So they're clonal, like the clonal expansion I showed you in the beginning. They're the same cell that's divided. And red is the exact same clone that appeared in 5 of the 6 people. That is, it had the same VH and the same V light chain, the same antibody, which is something very, very unexpected considering the diversity of the immune system. Why would 5 people from 2 different geographic locations have the same antibody? Next slide. Now this antibody we've characterized, it doesn't just react with Zika, it also reacts with Dengue 1. But it doesn't react with any of the other dengues, yellow fever, or West Nile virus. Next slide. Other antibodies that we cloned, not this expanded guy, had different patterns of reactivity. Next slide. And the antibody also neutralizes Zika, and it neutralizes Zika exceptionally well. So this is sub-nanogram or sub-nanogram IC50, so really potent. 100 times more potent than the best antibody we got to HIV. And even a day after infection, it's also working. Next slide. So these antibodies that are clonally expanded happen in 5 or 6 people. They're potent neutralizers, they protect, and they cross-react with Dengue 1. Next slide. So how come? Why is this happening, Dengue 1 and Zika? Well, so Albert has had a cohort for many years, and he was able to give us serum from before Zika and Zika. And we tested the serum against dengues, yellow fever, West Nile. And what we saw is that when Zika was introduced, well, you got increased reactivity to Zika, fine. But you also got increased reactivity to Dengue 1, but there was no Dengue 1 there at that time. So somehow the 2 of them are going together. So we asked, does having an antibody to Dengue 1 mean that you can react better to Zika? Next slide. And that's exactly what we found with a very, very high statistical significance. That is, people that had pre-existing Dengue 1 antibodies got better Zika. So next slide. So we wanted to know why. And the way we found out was by doing the structure. Structure of the antibody with Zika and the structure of the antibody with Dengue 1. And so 2 structures. And this is in collaboration with Pamela Bjorkman. And this is the footprint. The blue is the heavy chain, and the light blue is the light chain. And these 2 residues are contacted by both the heavy and the light chain. And you can see from this footprint why, in fact, you needed this one antibody, because both chains are contacting the Zika and the Dengue 1 in exactly the same way, actually. And they're using a very large part of the antibody molecule. Next slide. You can see that in detail here. The 2 antibodies are nearly superimposable. The Dengue and the Zika are nearly superimposable. Next slide. Even in the details of the structure and precisely the mechanism of binding is the same. Next slide. And it's a new kind of binding. So there have been a lot of antibodies to Zika and a lot of antibodies to Dengue, but this is something new. The way its mechanism of recognition is something different. And that's illustrated here. Next slide. So what we have here is an example of what immunologists call prime boost. Next slide. If you just get infected with Zika, well, you don't get much of a response, actually, because the virus isn't there for very long in most people. Next slide. But if you were primed to Dengue 1 and you have a pre-existing clonal expansion of the type that Burnett talked about, that cross-reacts with Zika, well, then you essentially get an anamnestic response, a memory response, but to the wrong thing. Not to Dengue 1, but to Zika. Next slide. So you start out with polyclonal B cells. Next slide. You get expansion of those cells, and then when you come back, you get a persistent response. Next slide. Okay, so start with this, and it turns out that it's interesting not only for HIV, but it's also interesting for other diseases. Next slide. Having cloned the antibodies to HIV, we have some insights into why it's in fact difficult to make a vaccine and why people have failed by and large. Next slide. It allows us to think about new ways of thinking about a vaccine. We don't have one yet. Next slide. And in fact, that way of thinking is probably correct based on model systems that we've developed. Next slide. It's true that learning something about the way people respond, especially these high neutralizers, gives us real clues about human immunity and also how you might think about specific vaccines, and also gives us reagents that we can use in experimental medicine. Next slide. And that's similar to what people have known for a very long time, over 100 years. Next slide. And it involves really getting the immune system involved as part of what we're trying to do. And finally, I wanted to show you a picture of Rockefeller University, which is where all of this is done. My lab is in this building here. We have a vaccine center here, which is also part of the lab. The hospital is absolutely tiny. We do all of the human work in that little tiny hospital. Only has 10 beds, but it's dedicated to human research. And so it really helps facilitate what we're doing. Next slide. And I wanted to acknowledge the people that were involved in the Zika project, which is actually just published. It's primarily Davide Rabaiani, but working with Charlie Rice, who's an expert in flaviviruses at Rockefeller University. The structural work was done by Caltech. We had collaborators in Mexico and in Brazil and at Yale University. Next slide. And then lots and lots of people were involved in the HIV studies that I talked about. So thank you very much for your attention and for the opportunity to visit Rio.
Okay, Glaucius, if you want to start. Glaucio, pode perguntar. And you are allowed to ask Portuguese. He will answer in English or Portuguese. You can ask in Portuguese. Glaucio?
No,
I think we can do it in English. First, congratulations, beautiful talk and beautiful work. My question about the sequential vaccination strategy. When you use a mutated or a challenge with a different bit of the antigen, mutated antigen, why doesn't the new antibody be specific to that one mutation and forget about the previous one? Or do you have to add sequentially all the different antigens together?
I don't know if this is on. Yes. So you don't have to add them all together. In fact, you don't want to do that. What happens is that as the antibody is developing, it retains the ability to neutralize the first virus, in fact. That's what has been seen in people. So there have been 4 of these studies done by different groups. Every time it's exactly the same story, essentially. The antibodies are developing, neutralizing additional viruses, but not losing reactivity. So at the end of this whole business, they're really very broad and neutralize many, many strains of virus.
Hi.
Yeah, well, congratulations. Yeah, very beautiful work. So concerning the Zika and Dengue virus, so there are some recent, I think there are at least 2 reports of ADE, antibody-dependent enhancement activity. So isn't there a risk that you may have some neutralizing for dengue and enhancement for Zika, or some specificity in causing, for example, in generating microcephaly or some of neurological?
So that's a very important question, and it relates to the fact that people that are infected with one strain of dengue, when they're infected with a second strain of dengue, have a higher risk for developing hemorrhagic disease. And that's been attributed to antibodies that are non-neutralizing but crossing between the different viruses. Now, in this particular case, the antibody only sees dengue 1 but not the other dengues, and it's highly neutralizing for dengue 1. So it's as potent a neutralizer of dengue 1 as it is of Zika. Whether this will cause ADE is highly unlikely because of its potency in neutralization, which is sub-nanogram. But, in fact, it has to be tested. Maybe it will protect against ADE, in fact.
Julio?
Hi, Michel. It's always a privilege for us immunologists to see you every 2 years and hearing about the new chapters of this incredible story. But anyway, just in relation to the macaque experiment, you emphasize that CD8 is important, but you didn't tell us that you are actually depending on the CD8 in order to generate, again, antibodies that have the same structural features. I mean, do you generate this high mutation that you had on the mouse and the human system?
No, in the macaque, so in the macaque that's controlled after the antibody injection, you generate, the protection is CD8 T cells. Now in humans, I didn't have a chance to talk about it, I just don't have a chance. When we do this experiment in humans and treat infected people that are viremic, we do see that their own antibody responses broaden and become more potent. Again, like the macaque, we don't really understand that, but it's a phenomenon that we've seen in people.
Okay.
Marcelo, just in front.
Hi. I really have two questions. One is the following. If in the neutralizing antibodies of Zika patients after they had dengue, they are equally highly somatically mutated as the...
They're not very highly somatically mutated at all. In fact, they are very similar to the typical human antibody to anything, which is 10 or 15 mutations, not 100 mutations.
Okay, so the second question is to produce broad neutralizing antibody.
It's the same epitope, right? So it's identical epitope, so there's no reason to repeat the germinal center and remutate. You already have a high affinity antibody. All you're doing is making it go.
No, this I understand, but is this a characteristic of the individual? Let's say the one that produces highly neutralizing antibodies against HIV, he will produce also highly neutralizing antibodies against influenza.
You know, we don't know that. Certainly for Zika, that's not the case. So for Zika, when we first did this, we first did it with the Mexicans, okay? And they were from a small village in the south of Mexico. And all three of them had the same expanded clone. And I said, well, they're brothers. You know, they must be brothers from some, you know, related mother or something. And so, but it's not the case. It's just because the people in Brazil have exactly the same antibody. So that's not genetics. That's not likely to be genetic. In HIV, it's not clear what it is because it's sporadic.
Okay, thank you.
So we use, does that happen, I mean the diversity in antibody repertoire, does that happen in other chronic infections? And does it happen in autoimmune diseases where you have autoantibodies?
This level of mutation is very unusual. I don't know of another circumstance where it happens.
Could you please comment on the breadth and potency of anti-HIV antibodies in the handful of HIV cure cases?
Oh, the cure cases. You know, there's no evidence that the antibodies are involved in those cases.
Elena?
Yeah, thank you very much for your wonderful talk. I was wondering if you could speculate what would be the structure relation between Zika and Dengue 1, because it's curious, because the disease here in Brazil, if you look at the person that had Zika 1, for instance, and it happens to be unlucky enough to get another Zika and Zika 3 or 4, there's going to be a tragedy, just the opposite.
So your question is about the structure of dengue and Zika and whether they're... So what the structural biologists, what we did together is show that in fact there is a region of the two that is so highly similar. It's a region actually that's involved in binding to the cellular receptor. So that is the site of interaction with the cell.
Arginine.
Yes.
Yeah, okay.
And that is conserved between the two, and it's so conserved that the antibody that sees one will also see the second.
If no other question, thank you very much, Michel. Very good. Thank you.